Hey everybody. So this video is going to be discussing contraction and relaxation of the muscle fibers while also including all of the action potentials and the roles that the nervous system is going to play in the contraction and relaxation of the muscles. So keep in mind everything we talked about in the previous videos. This will be kind of a sum up of all of those things, and let's jump right into it. So excitation is going to be when the action potential of the nerve fiber is going to tell the muscle fiber to contract, right? It's going to be excited. It's going to be told to do something. It's going to be able to contract. So the action potentials in the synaptic knob, I'm going to go between the images. In the synaptic knob, right? These action potentials in the synaptic knob are going to trigger the release of acetylcholine. Again, acetylcholine is the neurotransmitter that are located in the synaptic vesicles of the synaptic knob. So again, here we are. The synaptic vesicles are these little bubbles that surround the neurotransmitter, which is acetylcholine. Once they get that signal, they're going to release them into the synaptic cleft, where they can then bind to the opposite side, to the motor end plate. Once those acetylcholine neurotransmitters bind to the motor end plate on the opposite side, bind over here, what this is going to do is the sodium ions it's going to activate the motor end plate to open up ion channels, which are going to allow sodium ions to enter the muscle cell. What that's going to do is reverse the polarity of the cell, and the section of this cell membrane is going to also allow potassium ions to rush out, and the polarity is going to be reestablished, meaning that at this end plate, the motor end plate, once the neurotransmitters bind here, it's going to activate sodium channels to open up. Sodium is going to enter the cell, potassium is going to exit the cell, and we're going to get a net positive charge that's going to reestablish the polarity. What does that mean? As we mentioned a minute ago, here's our polarity situation. At resting membrane potential, we are negative, right? We are away from the middle, we are more negative. What I'm saying is when an action potential occurs and these neurotransmitters are released and they attach to the motor end plate, they are going to open up channels, kind of like, again, this is not the best image of it, but I'll do my best, right? It's kind of like it's going to open up some channels on here, it's going to open up some passageways, and it's going to allow for sodium to enter. The more positive you get, the less negative you get, which makes sense. Now we're going to move backwards towards the middle until eventually we start to go positive. Another way to look at this is this image here on a graph. So we begin in the negative location. So we start negative, right around negative 70 millivolts. Right? When the neurotransmitters bind, I'm just going to keep repeating this. When the neurotransmitters bind to the end plate, the sodium is going to open up and flood into the cell. Potassium is going to exit, which allows for a net positive charge to occur. And this is going to be the beginning of the action potential. And that's what it's talking about here. So this end plate potential, EPP, the end plate potential of the opening of the sodium and potassium channels is going to allow for action potentials to radiate in all directions. So you're going to see multiple of these action potentials happening all throughout the cell. This wave of action is going to reach the T tubules of the cell and in the terminal cisternae of the sarcoplasmic reticulum, and this is going to then allow for the calcium to start to release into the cytosol or the sarcoplasm of the cell. So during this excitation contraction coupling of the action potentials, the muscle fiber is going to lead to the activation of the myofilaments. In other words, once this action potential reaches the sarcoplasmic reticulum, it's going to then release tons and tons and tons of calcium into the cell, into the cytosol of the cell. All right. Here we go now, the next section. So first, we talked about the very beginning. We talked about the neurons sending signals down to the synaptic knob. Then synaptic knob is going to release neurotransmitters into the synaptic cleft right here, little blue dots. From there, the neurotransmitters are going to connect to the motor end plate, the opposite side. Once that happens, sodium is going to be allowed to rush into the cell, which is going to make it positive, right? Once it becomes positive, it's going to finish the action potential, right? So now the action potential can happen. So once this section up here occurs, once you get that positive, you're going to have the action potential occur. This action potential is going to then move through the cell, reach out to many parts of the cell, and allow for calcium to be released into the cytosol. So now we're going to talk about calcium, and this is really important for muscle contraction. All right, we discussed this earlier. There are multiple components to the muscle fiber, the muscle cell. These components are actin, which are these light purple sections, and myosin, which are these dark purple sections. We mentioned in a previous video how myosin wants to connect to actin so that it can grab onto it and pull it. Sliding filament theory, right? Myosin's going to grab on and pull it, right? Here's the problem though. These darker regulatory proteins that we discussed earlier, troponin and tropomyosin, are in the way. They're not allowing for this to happen. But I did mention to you earlier that there are sections on the troponin that bind to something and allow for the troponin and tropomyosin to move out of the way. And that something is calcium. When calcium binds to troponin, what it's going to do is it's going to move the tropomyosin out of the way of the actin and allow for myosin to bind to actin. So it's not going to happen without calcium. Think of calcium kind of like money, right? You have these bodyguards, troponin and tropomyosin, and they are blocking the entrance to actin. Not allowed in. You give them some money, they'll move out of the way for a little bit. In this case, the currency is not money, obviously, because it's your body, but it is calcium. So calcium is going to bind. It's going to move the troponin and tropomyosin out of the way. And then it's going to allow for the actin and myosin to bind, and then contraction can occur. I just said a ton of things. So my suggestion to you is this. Listen to this video. Watch a video on YouTube that is going to show you exactly what I'm talking about, the sliding filament theory. And then I want you to rewatch this video to better understand what just happened. All right. And it's all stuff I've been talking about over the last few videos. Remember, repetition is key today. So if you have any questions on this, please let me know.